ಕರ್ನಾಟಕ ವಿಜ್ಞಾನ ಮತ್ತು ತಂತ್ರಜ್ಞಾನ ಅಕಾಡೆಮಿಯಿಂದ ಎಲ್ಲರಿಗೂ ಶುಭಾಶಯಗಳು. ಇವತ್ತಿನ ದಿನ ನಾವು ನೊಬೆಲ್ ಪುರಸ್ಕೃತರ ಬಗ್ಗೆ ಒಂದು ಉಪನ್ಯಾಸ ಮಾಲೆ ಶುರು ಮಾಡ್ತಿದ್ದೀವಿ. 2022ರ ಸಾಲಿನ ಈ 3 ದಿನದ ಉಪನ್ಯಾಸ ಶಾಲೆ. ಇವತ್ತು ಸೋಮವಾರ, ನಾಳೆ, ನಾಡಿದ್ದು ಮಂಗಳವಾರ, ಬುಧವಾರ 3 ದಿನದ ಉಪನ್ಯಾಸ ಮಾಲಿಕೆಗೆ ನಮ್ಮ ಎಲ್ಲ ಶ್ರೋತೃಗಳಿಗೆ ಸ್ವಾಗತ. ಇವತ್ತಿನ ದಿನ ನಮಗೆಲ್ಲ ಗೊತ್ತಿರೋ ಹಾಗೆ ವಿಶ್ವದಲ್ಲಿ ಯಾವುದೇ ವಿಜ್ಞಾನದ ಕ್ಷೇತ್ರದಲ್ಲಿ ಮಹತ್ತರ ಘಟನೆ ಅಂದ್ರೆ ಇಡೀ ವಿಶ್ವಕ್ಕೆ ನಮ್ಮ ವಾರ್ಷಿಕ ನೊಬೆಲ್ ಪ್ರಶಸ್ತಿಗಳು. 2022ರ ನೊಬೆಲ್ ಪ್ರಶಸ್ತಿಗಳು ಅಕ್ಟೋಬರ್ ಮೊದಲನೇ ವಾರದಲ್ಲಿ ಎಲ್ಲ ನಮ್ಗೆ ಗೊತ್ತಿರೋ ಹಾಗೆ ಅನೌನ್ಸ್ ಆಗಿವೆ. ಈಗ 3 ವರ್ಷದಿಂದ ನಾವು ಇದೊಂದು ಸಂಪ್ರದಾಯನ ನಡೆಸ್ಕೊಂಡು ಬಂದಿದ್ದೀವಿ. ಅದು ಕೂಡ ನಮ್ಮ ಸನ್ಮಾನ್ಯ KST ಸದಸ್ಯರಾದ ಪ್ರೊಫೆಸರ್ H. ರಂಗನಾಥ್ ಅವರ ಸಲಹೆ ಮೇರೆಗೆ ಇದು 3ನೇ ವರ್ಷದ ಉಪನ್ಯಾಸ ಮಾಲೆ. ಇವತ್ತಿನ ದಿನ ಫಿಸಿಯಾಲಜಿ ಮತ್ತು ಮೆಡಿಸಿನ್ ಈ ಎರಡು ಕ್ಷೇತ್ರದಲ್ಲಿ ಅಥವಾ ಮೆಡಿಸಿನ್ ಅಂತ ಹೇಳುವ ಈ ಕ್ಷೇತ್ರದಲ್ಲಿ ನೊಬೆಲ್ ಪ್ರಶಸ್ತಿ ಪುರಸ್ಕೃತರಾದ ಪ್ರೊಫೆಸರ್ ಸ್ವಾಂಟೆ ಪಾಬ್ಲೋ ಅವರ ಬಗ್ಗೆ, ಅವರ ಕೆಲಸದ ಬಗ್ಗೆ, ನೊಬೆಲ್ ಪ್ರಶಸ್ತಿ ಅವರಿಗೆ ಯಾತಕ್ಕೆ ಬಂತು, ಅದರ ಹಿನ್ನೆಲೆ ಏನು, ಇದೆಲ್ಲ ತಿಳಿಸ್ಕೊಡೋದಕ್ಕೆ ನಮ್ಮ ಒಟ್ಟಿಗೆ ನಮ್ಮ KST ಸನ್ಮಾನ್ಯ ಸದಸ್ಯರಾದ ಪ್ರೊಫೆಸರ್ H. ರಂಗನಾಥ್ ಅವರು ಈಗ ವಿಸಿಟಿಂಗ್ ಪ್ರೊಫೆಸರ್ ಸೆಂಟರ್ ಆಫ್ ಹ್ಯೂಮನ್ ಜೆನೆಟಿಕ್ಸ್ ಬೆಂಗಳೂರಲ್ಲಿ, ಮತ್ತೆ ಚೇರ್ಮನ್ ಆಫ್ ಬೋರ್ಡ್ ಆಫ್ ಗವರ್ನರ್ಸ್ IIT ಕರ್ನೂಲ್ ಅಲ್ಲಿ ಅವರು ಬಂದಿದ್ದಾರೆ ನಮ್ಮೊಟ್ಟಿಗೆ ಇರಲಿಕ್ಕೆ. ಅವರ ಸೂಕ್ಷ್ಮ ಪರಿಚಯ ಅಂದ್ರೆ ಅವರು ಡಾಕ್ಟರ್ ರಂಗನಾಥ್ ಅವರು ಅನೇಕ ಕ್ಷೇತ್ರದಲ್ಲಿ ನಮ್ಗೆಲ್ಲ ಖ್ಯಾತ ವಿಜ್ಞಾನಿಗಳು, ನಮ್ಮ ದೇಶದ ಎಲ್ಲ ಅಕಾಡೆಮಿಗಳ ಸನ್ಮಾನ್ಯ ಸದಸ್ಯರು, ಬೆಂಗಳೂರು ವಿಶ್ವವಿದ್ಯಾಲಯದ ಸನ್ಮಾನ್ಯ ಕುಲಪತಿಗಳಾಗಿದ್ರು, ಆಮೇಲೆ ನ್ಯಾಕ್ನ ನಿರ್ದೇಶಕರಾಗಿ ಕೂಡ ಕೆಲಸ ಮಾಡಿದ್ದಾರೆ. ಅನೇಕ ಅವರ ವಿಜ್ಞಾನ ರಂಗದಲ್ಲಿ ಅವರ ಕಾಂಟ್ರಿಬ್ಯೂಷನ್ ಬಹಳ ಮಟ್ಟಿಗೆ ಬಹಳ ಇದೆ. ಅದನ್ನ ಈ ಸಂದರ್ಭದಲ್ಲಿ ಅವರು ಕೂಡ ಹೇಳಿದ್ದಾರೆ, ಜಾಸ್ತಿ ವಿವರ ಕೊಡಬೇಡಿ ಅಂತ. ಅದನ್ನ ಕೊಡದೇನೆ several recognitions, awards, INSA Young Scientist Medal, Aryabhatta Award, UGC Career Award, YT Tatacharya National Award, Sir M. Vishweshwarya Vigyan Puraskar, Dr. Rajaramana Award, SL Hora Medal of INSA. ಸೊ ಹೀಗೆ ಅನೇಕ ಪ್ರಶಸ್ತಿಗಳಿಗೆ ಅವರು ಭಾಜನರಾಗಿದ್ದಾರೆ. ಅವ್ರನ್ನ ಈ ಸಲದ ವೈದ್ಯಕೀಯ ಮತ್ತು ಫಿಸಿಯಾಲಜಿ ಬಗ್ಗೆ ನೊಬೆಲ್ ಪ್ರಶಸ್ತಿ ಬಗ್ಗೆ ಮಾತನಾಡಲಿಕ್ಕೆ ರಿಕ್ವೆಸ್ಟ್ ಮಾಡ್ಕೊಂಡಾಗ ಸಂತೋಷ ಒಪ್ಕೊಂಡ್ರು. ಇದು ನಮ್ಮ ಸೌಭಾಗ್ಯ. With this we have audience from all over the state. These lectures will also be on the YouTube after the series are over. So with these words I once again welcome everybody. We also have Dr. Ramakrishna, former director of the Zoological Survey of India, as I can see in our audience now. Many others have joined by also Professor T. Shivanandappa, ಅವರು ಕೂಡ KST ಪ್ರಶಸ್ತಿ ಭಾಜನರು. ಹೀಗೆ ಅನೇಕ ವಿಜ್ಞಾನಿಗಳು ಮತ್ತೆ ವಿದ್ಯಾರ್ಥಿಗಳು, ಉಪನ್ಯಾಸಕರು ಎಲ್ಲ ನಮ್ಮೊಟ್ಟಿಗೆ ಇದ್ದಾರೆ. ನಿಮ್ಮೆಲ್ಲರಿಗೂ ಶುಭಾಶಯಗಳನ್ನು ಕೊಡ್ತಾ ಮತ್ತೊಮ್ಮೆ ಪ್ರೊಫೆಸರ್ H. ರಂಗನಾಥ್ ಅವರಿಗೆ ನನ್ನ ಧನ್ಯವಾದಗಳನ್ನು ಅರ್ಪಿಸುತ್ತಾ ಪ್ರೊಫೆಸರ್ H. ರಂಗನಾಥ್ ಅವ್ರನ್ನ ಕೋರ್ತೀನಿ. I request Professor H. Ranganath to deliver this talk on Nobel Prize 2022 Medicine Physiology that has been backed by Professor Svante Pavlo. Professor H. Ranganath, sir.
Thank you. Thank you, Professor Ayyappan, for initiating this tradition at the Academy, because after you took charge of the chairman of the Academy, you started many initiatives. One of them is to organize public lectures of that current year on the Nobel laureates. So as you said, this is the 3rd year, and I am extremely thankful to you for inviting me to share my understanding with regard to this year's Nobel Prize on Physiology and Medicine. Thank you, Ayyappan, and also I extend a warm welcome to all the people who have logged in to participate in this discussion. So as you know, this year's Nobel Prize is given to a Swedish scientist, Svante Pääbo, for his contributions, for his discoveries on human evolution. And as you are aware, from 1901, it started the Nobel Prize awards. And now during 1901 to 2021, 609 times this award has been conferred on 975 people. Some of them got twice. Therefore, even though it is 609, there are 975 people are there. And of course, 943 individuals are being given. This is as per the Nobel Foundation. Now, I have gone through the citations of all these people who have been conferred the Nobel Prize. And to my surprise, for the first time in the Nobel citation, the word evolution has been mentioned. Therefore, I am extremely pleased and proud to say, for the first time, a Nobel Prize for evolutionary research has been conferred. And this, of course, many of our investigation research in biology and related subjects always related to evolution, in one way or another, as Dobzhansky said many times. Therefore, even in spite of that, for the first time, Nobel Foundation has recognized the original contributions for an evolutionary research, particularly which has enriched our knowledge on human evolution. And now, because as Dr. Ayyappa has already said, it is on the YouTube, and many people might have logged in who may or may not have the basics of this. Therefore, I will spend few slides on the preliminaries of this evolutionary concept, and particularly human evolution. As you could see here, we belong to a group taxonomically primates, and that appeared in a geological timeline 55 to 65 million years ago. Then 25 million years ago, our relatives apes came into existence, old monkeys, and 6 million years ago, humans and chimpanzees were found. And now, 3 million years ago, Homo erectus, our ancestor, that is hominidae ancestor, appeared 1.8 million years ago. As you could see here, from 55 to 65 million years, we have traveled down, we are 1.8 million years from our ancestor. And from this Homo erectus, he was in Africa, and then Homo erectus gave birth to another hominin that is called Homo neanderthalensis, our archaic relatives. And from then onwards, the preliminary Homo sapiens or hominins lived in Africa, and then they came out of Africa. In the meantime, our ancestor, Neanderthals, also was given birth by this Homo erectus. And as you could see now, the Neanderthal was first found in Germany, in Düsseldorf, in a valley. And their age is they appeared for the first time in the fossil record 400,000 years, and they existed till 30,000 years now. Subsequently, they disappeared, they became extinct. Anatomically, modern humans, that is ourselves, Homo sapiens, with skeletons like those of present-day humans, appeared first in Africa around 300 million years ago. Subsequently, around 60 to 70,000 years ago, migration from Africa into the Middle East led to the expansion of Homo sapiens. This is the famous out of Africa theory. That means the first Homo sapiens originated in Africa, then they migrated out of Africa and expanded to the Eurasia and other Asiatic regions. And now, during this phase, Homo sapiens coexisted with Neanderthals, because as I said, Neanderthals were in Europe. For the first time, their fossils were recorded obtained in Düsseldorf, Germany, Europe, and they were in Eurasia. And when Homo sapiens came out of Africa, they coexisted for nearly 20 to 30,000 years with Neanderthals. And of course, the nature of interaction, when they coexisted, when they're sympatric, Neanderthals and Homo sapiens are sympatric for some time in Europe, and even today it's a debatable matter. What was their relationship? What were their affiliations? Of course, all these things, what all I said to the timeline, for a long time, studies on human evolution relied on analysis of ancient bone remains, their morphological characters, and archaeological artifacts. this is the basis on which all these decisions have been made in the past. And as you could today, evolution is an extremely interdisciplinary discipline. And particularly human evolution involves several scientific disciplines. Inputs come different domains. For example, physical anthropology, evolution anthropology, primatology, archaeology, paleontology, neurobiology, linguistics, evolutionary psychology, embryo, like that, genetics, and goes on and on. That means to come to a message, come to a conclusion, a lesson about evolutionary history, one needs to have a comprehensive account. One needs to bring in the information from all these different branches of discipline to learn more and more about human evolution. And with all these things, as you could see here, there is one discipline which has emerged in the last 12 years, because of the contributions from this year's Nobel laureate, that is paleogenomics or paleogenetics. I'll come to that later. Again, to go back to the preliminaries, these are the people Darwin, most of you know, Charles Darwin and Alfred Russel Wallace jointly contributed to our basic understanding. And they gave a very unifying definitions in biology, that is descent with modification. That is, they defined evolution as descent with modification because they heavily depended upon the paleontological evidences and the then available evidences. And the message was, from all these evidences, the beings now living have descended from different beings which lived in the past with modification. That means we had ancestors, and these ancestors undergone modification, and the descendants were born. That means descent with modification is the mantra of a process of evolution. How it happened, what are the mechanisms, they did not know. But this was the unifying definition. Even today, descent with modification could be utilized at the level of molecular biology, even at the level of genomes. So right from the fossils with all the other evidences like comparative anatomy, embryology, paleontology, natural populations, all these things gave evidence to the descent with modification of Charles Darwin. And many a time, Darwin himself has said fossil record is incomplete because there were not many intermediate stages, but it gives us a clear proof that the existence of past forms of life, or whether it is embryology or comparative anatomy, it gives you one type of information. The fossils gives us a very specific information with regard to the existence of life before us. And as I could see, fossils, this is a book recently published, fossils sparked Charles Darwin's imagination to make generalization with regard to descent with modification, how it happened over a period of time. And you could see the process in the Darwin book, Origin of Species, you could see the procession of life, right from primitive fossils to the latest mammals. You can see how the procession of life has taken place from one primitive to the other organisms with modifications over a period of time. One basic message is all forms of life did not appear simultaneously. It came in succession, one after another, with modification. That is the philosophy of evolution. And now, when Darwin came out this theory of evolution, he was not aware of how the variability is there, etc., etc. And as you are all of you are aware, that with the rebirth of genetics or Mendelian principles, it was understood how the variability could be obtained through genes and mutations, and subsequently people tried to extend the Mendelian principles to the evolutionary phenomena of natural selection, etc., as advanced by Charles Darwin. But for a brief period, there was a conflict between the experimental biologists, that Mendelian geneticists, and biometrician quantification cytogeneticists. And later on, this conflict was resolved. And finally, they realized that Mendelians and biometricians' principles are compatible, and the statistical treatment of heredity was shown to be compatible with Mendelian rules. These are not two different things, they are compatible, and many people contributed to this gelling these two things, and the process of evolution and our understanding of the process of evolution continued. And now the basic things that have happened during this is, after there were many gaps in the Darwinian principle, Darwinian concept, that is, he did not know the origin of variability. Then with the rebirth of Mendelian genetics, people related genes and mutations. And even at that time, people did not know how genes are organized, how mutations are formed, how the variations are going to be produced. But over a period of time, all these gaps in our knowledge was filled up by the knowledge created by various scientists right from 1930 subsequent. Even today, people are giving us more and more information about how the variations are being formed, how the diversity is going to be established, and how this diversity is being maintained. And now what has happened is, as the resolving power of our understanding of the origin and maintenance of variability, sustenance of variability, the resolving power of our understanding is becoming more and more from Mendelian genetics to molecular biology and genome biology. Because of the inputs coming from various sources, our knowledge has improved, and many new concepts have come into existence, only to strengthen Darwin and to see new dimensions of the evolutionary process. And of course, as you are aware, again, one more preliminary, Hardy and Weinberg started the population genetics, and they gave us a formula, Hardy-Weinberg law of genetic equilibrium, to see how allelic frequencies are there in natural populations, and how allelic frequencies could be changed under the influence of selection, drift, migration, etc. That was the starting point of population genetics and evolution. Subsequently, you could see Ronald Fisher, Haldane, Sewell Wright, Huxley, Ernst Mayr, and Dobzhansky. Many more people contributed to the neo-Darwinism. That means whatever gap was there in the Darwinian philosophy was filled by these neo-Darwinists, and all these people contributed to the birth of neo-Darwinism. Even at this stage, we knew only the variability, the allelic frequency, and the different strategies of natural selection, how it is being maintained, and alternatives, things like the genetic drift, sexual selection, etc. And the important gap at this particular point of time was the people did not know the molecular anatomy of the gene or the molecular basis of mutation and other things that are related to the variability. And subsequently, as you could see now, a host of our doyens of molecular biology, light from Watson Crick to Wilkins, Chargaff to Corona and others, all these people contributed in their own ways to understand the chemical basis of the gene, molecular basis of the gene, how it exactly acts, all the transactions of the gene, like structure, function, transcription, translation, the molecular basis of mutation, molecular basis of repair, recombination. All dimensions of nucleic acid transactions were resolved by these people. With the knowledge coming from all these people realized how variations could be understood at the organismic level, at the phenotypic level, at the molecular level. And this was a real breakthrough in our understanding of evolution, and particularly the mechanisms underlying evolution. And with this, as I said already, population genetics, the Hardy-Weinberg, then the neo-Darwinism. Now, descent with modification was Darwin's definition, and now Hardy-Weinberg law and the follow-up theories that any change in the allelic frequencies could be treated as evolution. So people try to understand and give us how exactly allelic frequencies could be changed from generation to generation, etc. So people recognize the evolutionary forces like varied strategies of natural selection, genetic drift, migration, competition, altruism. All these things could contribute to the variations in the allelic frequencies. That means Hardy-Weinberg equilibrium is removed. There is no longer equilibrium. Always it is in a dynamic phase, and that is the process of evolution. And now with the structure and function of the gene, people now know the molecular basis of mutations, and also people now realize that molecular basis of genetic variability, and throughout the genome, people could see variabilities in all parts of the genome. It can be structural genes, it could be regular regulatory gene mutations in the repetitive sequences, all types of, the entire genome is susceptible for variability. Some variability can influence or reduce the fitness or can improve the adaptation. There are many, many mutations, many, many variabilities in all of us, from bacteria to man, which apparently may or may not contribute to the fitness, and even them they are being maintained. Therefore, there was a strong interaction discussion how the neutral mutations, so-called neutral mutations, are being maintained, how this variability, who is guiding this neutral variability. Whereas if a variability is influencing the adaptation, etc., it is under the influence of one or the other types of selection. Whereas if it is a neutral, if it is not influencing any phenotypic or adaptive phenotype, and there was a problem. And then many theoreticians, population geneticists, molecular geneticists contributed to the neutral theory of evolution. That means we have deterministic forces and also stochastic processes in evolution to decide the genetic variability, to take care of the maintenance of the variability. And along with that, new phylogenetic techniques. Phylogeny is important because you may have variability, and this variability has to be linked from one sequence to the other. Then only it is possible to give some meaning, some importance to this thing. Therefore, people invoked new techniques to construct evolutionary trees, and the most important springboard in biology, and particularly to evolutionary biology in terms of this year's Nobel Prize, is the genome biology. And as you know already, the success of the Human Genome Project encouraged many more genomes to be sequenced, and today hundreds and thousands of species of genomes are available, from microbes to man. And because of the information available, new fields like structural, comparative, functional, evolutionary genomics are in place now. Earlier, all these things were there without genomics. Evolutionary comparison of fossils, etc., etc. Structural comparison of the morphological character of the fossils or anatomical features. But today, same comparisons could be made with the data available from the genomics. That was the springboard, and from this, as you could see later, a paleobiology or paleogenomics took off. And voluminous data is available from the genome projects, and now it needed bioinformatic tools. Therefore, interpretation and usefulness of genome sequence data for addressing evolutionary questions, we need new logarithms, new bioinformatic tools, and that also helped them. So the springboard for future evolutionary biology, particularly paleogenomics, paleogenetics, is genome biology and the bioinformatic tools. So the stage was set with these things. The techniques were available, so isolation of the genome, sequencing of the genome, and getting the information out of that genome through these things. Because of the template was ready, it was easy for the subsequent people to get into the bigger questions in evolution now. And now people are using all this information to extend evolutionary synthesis. Even in India, there are many labs who are contributing to the extended theory of evolution by using all these things, including theoretical models. And now that was the information I wanted to share with you before I take up this year's Nobel Prize. 1859 book Origin of Species, as Charles Darwin, particularly the fossils, he treated as documents of evolution, and it gives us the direct record of life over geological time. That's why he said it is a documentary. And now, coming to this year's Nobel Prize, 22 is awarded to scientist from Sweden. And what is his contribution? His contribution is modern humans, we the modern humans, Homo sapiens, share DNA with extinct homin relatives like Neanderthals and Denisovans. The entire story, what I'm trying to share with you in next few minutes, half an hour or so, revolves around how modern humans, we share our DNA, share our genome with our extant relatives like Neanderthals and Denisovans. So the Faber's contribution is to get the genomes of Neanderthals and Denisovans and comparing those genomes with the existing extant genomes of the modern humans. And let me go to that now. First, who are Neanderthals? Because we don't know, people may not know who are Neanderthals, what is the importance of them, etc. And as I said already, Neanderthals have different names: Homo neanderthalensis, Homo sapien neanderthalensis, and these are the extinct archaic humans, and they emerged at least 200,000 years ago during the Pleistocene epoch. That means about 2.6 million to 11.700 years ago. That is the geological time. And now the name Neanderthal, who gave this name? This Neanderthal name is derived from the valley in Germany, in Düsseldorf, where the first fossils were discovered. The first fossils were discovered in this valley in Germany. That's why they called it as Neanderthal or Neanderthal, Homo neanderthalensis. In fact, I was fortunate when I was in Düsseldorf, visit this valley. So how people are excited to visit that. It was a tourist attraction in Düsseldorf to see how fossils are organized in this particular round. Of course, after the first discovery in Germany, and these fossils have been discovered in other parts of Europe also. The most recent fossil and archaeological evidence of Neanderthals is from 40,000 years ago. In Europe, after that point, they became physically extinct. Please note the word I have used. After this, we did not have a fossil representative of Neanderthals. But what happened now is, even though they are physically absent, a part of the genome, a part of the DNA is with us today. This is the contribution of this year's Nobel Prize winner, Savante Barrow. So that's why when we use the word extinct, we have to be extremely careful now. Physically, they may be absent, but we have inherited with some modification, descent with modifications, the genome of this Neanderthal, and we carry them in all over the world now. And this is the distribution, as you could see here. And this is the Neander Valley, where the first fossils were discovered, and subsequently now many more fossils were discovered in other parts of the globe. And as I said, they are no longer, we get fossils beyond 40,000 years ago. And one important thing that what we have to remember now is we have a paleoecology. That means by looking at the fossils, one can infer certain of the things about the paleoecology. That means possible circumstances, ecological conditions, how they lived, where they lived. Neanderthals lived before and during the last ice age of the Pleistocene, and unforgiving environment. That means actually the freezing environments, and very rarely it is inhabited by humans. And the very fact they survived in such environments, people infer their survival during tens of thousands of years of the last glaciation is a remarkable testament of human adaptation. They did not know the mechanism of this adaptation. They did not know how they adapted it, but the very fact they survived in such regions, the very fact we are getting fossils in those regions, tell us that they were adapted to live in this condition. And later on, you will realize at the end of my talk that there are a few genes which confer them to survive, which provided some adaptive value for them to survive in such environments. And at least in Tibetans, the remnants of those genes have been traced recently. And this is the fact of Neanderthals, 400,000 to 40,000 years, and the weight, most, of course, physiology can also help us to know what they were eating, etc. These are some of the things that people have cataloged with regard to the Neanderthals. And as this is the bits and pieces of the skeleton of Neanderthals, which was obtained in Germany in that particular valley. And as you could see here, until the late 20th century, Neanderthals were regarded as genetically, morphologically, behaviorally distinct from living humans. This was the belief, but this is changed today. Genetically, we are related, morphologically, we are related, and all these things are modified through genomic analysis now. And now, this is the most extraordinary serendipity in science now. Nobody thought, or even the Pavos group never thought, imagined that they are going to have this discovery. And now it is a distinct group of hominin. And the fossils were recovered in Siberia, Altai Mountains in Siberia, because now they are looking at the Neanderthal fossils in different parts of Europe. And when they were analyzing, trying to excavate the fossils from in the mountains of Siberia, they got a bunch of bones. And from this bunch of bones, they analyzed the mitochondrial DNA as well as the nuclear DNA. And now, of course, he was a PhD student, Johannes Kroes, and he was extracting the mitochondrial DNA, thinking that it may be a Neanderthal bone. It was a single bone which was available, and he was trying to understand the genomic organization of this particular bone. And now, of course, later on, I'll come back to this. The Krauss realized that it is not Neanderthal bone because the mitochondrial sequence was different, the nuclear genome sequence was different, and he stumbled upon a new lineage, and they called it as Denisovans, based on the cave from where it was discovered. And of course, they first appear to have entered the stage from perhaps as early as 287,000 years ago. Of course, this is the dating of through the fossils and also through molecular genomic sequence using molecular clock phenomena. They say these Denisovans, they were discovered later, but they have appeared 287,000 years ago. And this is the cave. And this is the cave from where this bone was recovered and subjected to analysis, and the data revealed that it is distinctly different from the Neanderthals, and they named it as Denisovans. And this is the bone. This simple bone gave a recognition to a new hominin, and that new species is called the Denisovans. And I have modified the scheme got from Wikipedia. As you could see here, this is the common ancestor Homo erectus. Then it gave from Heidelbergensis. This is the next stage. From Heidelbergensis, there is further descent with modification, giving rise to Homo sapiens. These Homo sapiens and their relatives, ancient sapiens, lived in Africa. At the same time, the two more sister relatives emerged from this ancestor. One is Neanderthal and another is Denisovan. Neanderthals were in Europe, Eurasia, whereas Denisovans in Russia. So like that, two branches were there. So now the contributions of Pablo is around the genome of Denisovans, genome of Neanderthals, and comparing it with the Homo sapiens. They were in Africa, these people were in Europe, and this out of Africa, as you people know, that Homo sapiens started coming out of Africa, and they colonized the Eurasia, and then they moved to different parts of the globe. That means when they came out of Africa, already there are the representatives of Neanderthals and Denisovans, and they coexisted for a certain period of time. And this is the most crucial information. This contribution has come from Pablo now. So now I will concentrate only the contributions with regard to this particular three people, and what is the outcome, what are the lessons we have learned from this particular study. Now, the paleogenetics or paleogenomics, basic definition could be discipline of paleontology with its fossil records as documents of evolution confirms the existence of now extinct species and in a few cases captures potential in between forms of the modern species. A paleogenome is the genome of an extinct organism. For example, the genome of dinosaurs, Neanderthals, or extinct plant. And this helps us to get a glimpse of DNA of the past. So here I must say, fossils, as I said many times already, fossils were the documents of evolution. Fossils gave us morphological characters. Characters were many things for decision on descent with modification. And fossils were used for different people from different processes by paleoecology, for example. Sometimes even paleobiochemistry people, paleophysiology people used the fossils for their analysis. But one inaccessible part of the fossil, which was not available to the earlier evolutionary biologists, is that those fossils, those bones, those teeth had DNA in them. It was an inaccessible part. And now the techniques are available to extract the DNA from these fossils and to study further now. To do all these things, as I said already, that the template was ready. The molecular biologists have laid the template for isolation, sequencing, and all the techniques were available. And also the informatic tools were available now. And now, as I said, I emphasize the inaccessible part of the fossil is made available, and that is called the ancient DNA. And Pablo and his group and many more people contributed to study this ancient DNA, and many new decisions, many new things, many new questions could be asked, which we were unable to question, unable to imagine unless this was available. Ancient DNA. For the first time, DNA from fossil specimens are being used to learn and uncover evolutionary phylogenetic lineages. For the first time, all the time, fossils were used through different sources. And in this direction, the important challenge was to get an uncontaminated, pure genome from fossils. And all these techniques were available, as I said earlier, isolation of DNA, sequencing of the DNA, using the informatic tools, analyze the genomic sequence, and many things can have been studied of the extant animals, living plants, living bacteria. But now people have to take up the challenge of getting the DNA from the fossils, that too uncontaminated and not destroyed, not subjected to post-mortem changes in the pure form. That was the challenge. And this is the timeline. I will not go into the details. See, this paleogenomics, this paleogenetics has a history of 40 years now. It all started in 1984, and even today it is being fine-tuned. And this is the timeline given by in the review article. And the first so-called ancient DNA was isolated in 1984, but it has a lot of contaminations, other things. It was from a zebra belonging to the family Equidae. Like that, parallelly, you could see the laboratory on this side, how the laboratory techniques were fine-tuned, and in every stage, the dilemma was how to improve and get a puriform form of the genome. And this art went on, and by 2017, you could see an automated machine was available to capture the ancient DNA. But this was a struggle for nearly 30 years, defining the process protocols to make sure uncontaminated, undestroyed the genome to be available. Simultaneously, these are the dry laboratory techniques. That is the particularly most of you know, with the PCR, it revolutionized our genomic studies, then next generation sequencing and modifications of this next generation sequencings are available, and correspondingly, many bioinformatic tools are available. So in combination of the laboratory methods and the dry laboratory advances put together was available now, and for this, major contributions have come from the Pablo group. That in recognition of that, he was been awarded the Nobel Prize. So I will not go into the details of all these stepwise success stories, because what I'm interested, what people are interested is, what is that new information that people got from this discovery? And now, in brief again, so ancient DNA is subjected to many changes, postmortem changes. For example, in another review, you could say oxidative damages could happen, cleavages can happen, and degradation of the material, DNA crosslinks, hydrolysis, all these things are expected to happen and damage and destroy the DNA. And in spite of all this, you must be in a position to do the technique like PCR, multiple PCR independent cloning, then the library, building the library, and authentication of the library through different investigations. So my only message is the Pablo and his group took care of all these things. The hallmark of the saga is at every stage, methodological improvements were made, particularly sequencing methodologies, and now new algorithms, new models are available to analyzing and mapping the data now. Huge data is available, and this data of the fossils have to be compared with the like existing chimpanzees, the apes, and the Homo sapiens. And now the most important thing is, at every step, care has to be taken to avoid contamination due to microbes or endogenous or exogenous. For many sources, the fossil DNA could have been contaminated, and that has to be taken care of. Repeatability of the results. This is crucial, and I must say here, Pablo and his group repeated the results in their own lab, and also they shared the samples to a few other labs in US and Europe to see whether the results are repeated. So they got the authentication, and also they could see repeatability of the results. They move on and on. And again, one more way of authenticity is repeatedly they compare the data coming from the fossils with the existing genomic sequence of the Homo sapiens, the chimpanzees, and other known sequences. So that's why it took 10 years for them to standardize all these things to say that extinct genomes from extinct hominins can be extracted, and it can be used to study and ask many more new questions in evolution. And when I gone through the findings of these people, and as I could see here, the challenge was to get uncontaminated pure genome from fossils. The Nobel laureate of this year and his group struggled for over 2 decades and succeeded in establishing a protocol to extract from the fossils. Modern humans share DNA with extinct hominin relatives, and this was clearly established in the last 10 years. And once this was published, now you could see very alternative issues of the journals of science and nature are getting more and more information about how the genome is shared between humans and our extinct relatives. And now, an important message I could get from this story reflects commitment, dedication, and perseverance of scientists, unperturbed by failures. There were many failures during these 10 years. And most important thing for me is collaboration and cooperation among different laboratories. When Pablo started the analysis of the genomes of the Neanderthals and Denisovans, he got the specimens from different parts wherever the fossils were available. And openly they shared the fossils of Neanderthals, particularly Neanderthals, to the Pablo, and Pablo collaborated with them and made joint publications, and also during cooperation, they authenticated the results. And the most important, fossils are very precious. They are not available in every lab, and wherever it was available in different parts of Europe, he could establish collaboration and with cooperation, they shared the data and it was authenticated. And as I said, it was a collective effort of many labs, and finally the bonus was on him to extract the DNA and share with that. And now the genomic sequences are available. And now the next question is how to make use of these sequences that are available from the extinct animals, particularly Denisovans and Neanderthals, and how it could be useful to study the evolution. And now some of the questions could be discussed all along, like introgression and hybridization in evolution, because even in Darwin, in his book, one last chapter is hybridism. He has realized the importance of hybridization between species to generate more variability and its impact on evolution. And it's a constant evolutionary catalytic force to generate new variabilities in permutations and combinations getting from two different species. And from this, you can also switch on to discuss the concept of Ernst Mayr biological species concept and also the phylogenetic species, because biological species concept emphasizes on reproductive isolation, whereas phylogenetic species concepts emphasizes on the phylogenetic history. You recognize the species based on its uniqueness, based on its ancestors, and reproductive isolation may not be the criteria. And also by comparing the genomes of the past and the present, you could see the evidences are there at the genetic level of positive selection or selection pressures. And now, what makes humans human? The genes that make us human are what makes uniquely human. Because all of us, now I say, their ancestors, Denisovans, the Neanderthals, are all relatives. And now this comparison of the data tell us what are the genes, what are the characters which make us really human. And the most important message I could get from all this study is the genes themselves may be older than the species that bear them. We have many genes in us, in the genome of human. Homo sapiens have many genes, many clusters of genes. But the age of those genes are much older than the age of the Homo sapiens. It's not only true for Homo sapiens, but many other species now, because genes are carried from ancestors to the descendants. The genes of the ancestors are thriving in us wherever they have been selected and with some modifications. So basically, some of the Neanderthal genes, Denisovan genes are with us. That means some genes of us are older than what we are. And this genome analysis helps us to show how migration has taken place. And at every stage, you can take up the discussion of descent with modification. Darwin emphasized descent with modification to the characters which are visible to him. Today, descent with modifications could be discussed at the level of genomes. What are the common sequences and what are the sequences which we call them archaic sequences, and how these archaic sequences of Neanderthals and Denisovans are modified in humans, etc., etc. So that's why I said descent with modification definition given by Charles Darwin is the most unifying definition. One can extend it to all levels of our understanding depending upon the expertise available with us. Of course, we can ask many more questions. And now the first thing is, I am bringing the contributions of Alan Wilson, because after PhD, Pabo became a postdoc in Wilson's lab. By the time Pabo became a postdoc, Alan Wilson already has studied the mitochondrial DNA from the current African or non-African populations. So it's not a fossil, it's not a paleogenome, but already has analyzed the mitochondrial layer of the present African populations and also non-African populations. And you could see the difference. His results indicated common origin in Africa for all the subpopulations of Homo sapiens. That means out of Africa theory got the boost from this finding. Homo sapiens, first they were in Africa, then they migrated to Eurasia and they spread. So African populations even today have mitochondrial DNA whose sequence slightly differs from that of non-Africans. And that's how already he has established this line of study at that point of time. Santa Paolo, after his doctoral degree in Sweden, he joined as a postdoc, and for first 5 years, he spent on retrieving, sequencing, analyzing the DNA from archaic species. So that was his focus as a postdoc. And Pabo and his collaborators focused on the Neanderthal genome that was extinct, and they also they tried to get many more fossils, but it was not possible. So they restricted themselves to Neanderthals to start with. And again, the first point is mitochondrial genomes. But is the postdoc mentor, Alan already studied the mitochondrial genomes of the present, and now he wanted to look at the mitochondrial genomes of the Neanderthal fossils. Why he picked up mitochondrial genome is, as you are aware, there are many copies of mitochondria in every cell, and multiple copies of the genomes are available. And also, second thing is, the mitochondrial genomes are not subjected to meiotic recombination because there are many advantages. Simultaneously, there may be many disadvantages. Let me take up that later. So mitochondrial genomes were analyzed in these Neanderthal fossils. And as I said, he could collaborate from Germany, India and Croatia, and also various labs wherever the Neanderthals were available in different parts of Europe. And he compared that sequence with Homo sapiens, with a collection of 2,000 odd human and 59 chimpanzee. That means he could get all this information from other labs. So he focused on the mitochondrial DNA of Neanderthal fossils, and the data, whatever he got, he could compare with that of the chimpanzee and Homo sapiens. And the most important here is that he got the fossils from different parts of the Europe. That is the most, and people openly, wholeheartedly shared with this. And the message is the sequence was shown to fall. The sequence of the mitochondrial DNA of Neanderthals fall outside the European, Asian, Native American, and Australian, and Oceanic Homo. That means it was distinctly different from all these different Homo sapiens now. So Neanderthals fossil DNA was compared, and he could show that it was totally different. And he came to the conclusion that Neanderthals may not have contributed much to the mitochondrial DNA of the Homo sapiens. Pabo, now new techniques were available to construct phylogenetic trees, and the mitochondrial DNA of modern humans originated in Africa, that is Homo erectus, carrying the mitochondrial DNA that lived 120,000 to 150,000 years ago. Based on the mitochondrial analysis, the time since the existence of common ancestor of Neanderthal and modern human. So here the message is both Neanderthal, as I said already, both Neanderthal and anatomically modern human had a common ancestor. And that was the age of that common ancestor is about 550,000 years to 690 years. The results suggested that Neanderthal went extinct without contributing mitochondrial DNA to the present humans. So both Neanderthal and the anatomically modern humans have a common ancestor, and after that, there's a divergence. Mitochondrial DNA of Neanderthals appears to be different from that of the Homo sapiens. And after mitochondrial DNA, because he could realize that if you depend exclusively on mitochondrial DNA, you may not be able to get the total information of the genome. Then he switched on to the nuclear genome. And now again, he need to have more bone samples. So 17 Neanderthal bone samples were got from different labs across Europe, like other countries. And of all these samples, what he got, there was one unique fossil from Vindija cave, Croatia. And this fossil from Croatia has a mitochondrial DNA. 90%, but it was similarity to the mitochondrial DNA of the Neanderthal. Already I have said Neanderthal mitochondrial DNA is a unique. And now, before getting onto the nuclear genome, Pabo and his group looked at the mitochondrial DNA of all these samples, and of all the material, the fossil from this Pindija cave showed it has a mitochondrial DNA, which is almost 90% is similar to the Neanderthal information already they have. So they took the nucleus, they took the nuclear genome of the fossil from the Vindija cave, because Vindija cave, now it's very clear that it is of a Neanderthal man individual, and now because they wanted to analyze the nuclear genome, they picked up this particular individual and isolated the DNA and they sequenced it. And now the challenge was much more different, and as I could see in his news article publishers, his own group alone cannot do the entire analysis of the genome that is coming now. So at that point of time, it was an extremely interdisciplinary research, and also he wanted to have more bioinformaticians, genome scientists, etc. He says he has to bring in the cooperation and the collaboration of 50 scientists with different backgrounds. It's the huge, of course, they are analyzing the fossil DNA, and he established a team of 50 different scientists to uncover the findings. And these are the bones of the Vindija fossil, which undoubtedly it is of Neanderthal, and now based on the mitochondria, and now they analyze the DNA of the genome from these bones, and these are the places in Europe from where they got these samples, and this is the one which give them the sample for genome analysis, which was unequivocally of Neanderthal origin. And now they analyzed the nuclear genome of this particular individual, and in 2010 they published this material, and groundbreaking publication. Neanderthals, the closest evolutionary relatives of the present-day humans, lived in large parts of Europe and Western Asia. Of course, they disappeared 30,000 years ago. The draft sequence of the genome was organized, and it had about 4 billion nucleotides, and the sample was from 3 individuals. From 3 individuals, of course, the consensus library was established, and it had about 4 billion nucleotides. You may have a question, human genome has only 3 billion, how it come 4 billion? But the initial analysis showed it has. And again, the samples were analyzed from Spain, Germany, and other caves, Russia, and all these nuclear genomes coming from various samples of Neanderthals confirmed or almost their equivalent to Vindija individuals. So Vindija Neanderthal individual sequence was compared with the Neanderthal sequence coming from different sources. Authentication was done, and finally they could establish the draft sequence of the Neanderthal genome. The Neanderthals from across great part of the range of the Western Eurasia seem to be equally related to present-day human. That means all these Neanderthals, they are of the same type based on the nuclear genome, and their relationship to the humans is almost to the same degree. And this is the original publication. As you could see here, a draft genome was published in 2010, and so many collaborations. I have not taken the addresses of all these people. It's a huge list and reflects collaboration and cooperation. The analysis of the Neanderthal genome, they are likely to have had a role in the present ancestor, the present-day humans outside Africa. One of the implications results in point to a number of genomic regions and genes as candidates for positive selection early in modern human history. And of course, I will come to that later. That means the preliminary information of this draft sequence organized by these people showed that it has a lot of impact on the human genome of the present species. At the same time, they could see that the data by comparing the present day extant genome of humans with that of the Neanderthals, nearly 1 to 4% of the genomes of the people in Eurasia, Homo sapiens, are derived from Neanderthals. So they could compare with appropriate software the Homo sapiens genomes of the Eurasia and the Neanderthal sequence. They got it from the bones, and depending upon the regions where they got, nearly 1 to 4% of the genomes are same. That means these people have inherited nearly 4% of the genome from the Neanderthals. Further analysis identified variants that occurred in all sequenced present-day humans, while they were absent in Neanderthals. So 1 to 4% of the genome was of the Neanderthal, and subsequent analysis of the human genomes from different parts of the globe showed these archaic genes have some variations over a period of time now, and all these variations are unique to Homo sapiens, and they are absent in Neanderthals. This is again a clear example of descent with modification. The 4% of the genome were derived from the ancestor Neanderthals, but later on, many variations have taken place because of mutations, and these variants are present in different individuals across the world, while these variants are not found in Neanderthals. So we know the ancient sequences, archaic sequences, and also the derived sequences. That's how the comparative genomics has helped us today. And now, as I already told you, a serendipity has happened, and now another relative Denisovan genome is available. Again, they subjected the Denisovan bones, which I already shown you, and they isolated the mitochondrial DNA from the Denisovan and also from the nuclear genome, and they could show the mitochondrial genome and the nuclear genome of the Denisovan finger, which was obtained in that cave, was totally different from both Neanderthals and Homo sapiens. Fabulous group discovered entirely a new hominin now, distinct from Neanderthals and Homo sapiens. Based on the nuclear genome, based on the mitochondrial genome, they could show the bone from the cave, what they got, has a sequence which is distinctly different from the Homo sapiens and Neanderthals. So for the first time, this is of course, they got some teeth also. This is the bone, and these are the two important publications that have come in Nature and Science. It showed for the first time a new species have been discovered. Not looking at the morphology and other characters, just by looking at the DNA sequence. In addition to bone, they also got 3 teeth. And the fabulous team carried genomic sequences of these teeth from the cave. They are highly similar, but distinct from the finger bone. They have the genomic sequence of the finger bone, and in the same cave, in addition to bone, they got some teeth. And they analyzed the genomic sequence from this teeth, and they could show the mitochondrial DNA is same, but the genomic sequence was different. And from them, they concluded the teeth and the finger bone are likely to be derived from different individuals. So like this, of course, you may have questions with regard to the sample size. Fossils are very few in number, and these are highly precious. Wherever they got this information, they could show that Denisovans have an evolutionary history distinct from Neanderthals and modern humans. This was the foundation. Said, see what happens next now. This is the publication. Furthermore, the discovery had been entirely by retrieval and sequencing of archaic DNA without any morphological information. It's very rare. Just looking at the DNA sequence, they have identified a new species whose full body is yet to be recovered. Sometimes I compare this vision of Pavo and his group to the periodic table. When periodic table was organized, he left some gaps hoping that with that atomic weights and atomic number, metal will be that later on those gaps were filled up. Similarly, here they expect sooner or later you make a full skeleton of Denisovans, and now in the absence of that, they could have defined a new species. So this is the modern humans. Now this is the data that is available, and now modern humans share DNA with extinct hominin relatives, Neanderthals, and then even evolution, and further discoveries have been made. And this is possible because of comparative genomics. Paleogenomes were compared with the extant genome is Homo sapiens. This was the contribution, and it has a lot of implications now. How do we know that the genes have come from Neanderthals or Denisovans? And now the genomes of several Neanderthals have been fully sequenced, and these genomes have been compared with the modern day human sequences. And now people have compared the people from sub-Saharan Africa, which contain absolutely no archaic genes, as this population was never existed in Africa. So though people lived in Africa, came out of Africa, and then mixed with Neanderthals, and they spread over it. Whereas these people were never existed in Africa, they never show the symptoms of archaic genes present in Neanderthals. And now, since the human population migrated out of Africa, once hybridization occurred with Neanderthals, the population spread outward to Europe. That is what has happened. Whereas these people in Sahara Africa, they never had this opportunity. Therefore, Homo sapiens have the remnants, the relics of Neanderthal genome or the archaic genes, while these people did not have. Human populations in Europe, Asia, and Oceania contain highest percentage of archaic genes, but of course the percentage is small. And now the most important thing, consequence of all these things, people recognized in breeding the genome of an offspring of a Neanderthal mother. So this is the first ancient human hybrid, a hybrid between Neanderthal mother and Denisovan father. Again, one or two individuals are obtained. That was published in 2018. So by 2018, the genomic sequence of Neanderthals, Denisovans are available. And now when this fossil was available, they never knew that is a hybrid. But later on, the nucleotide sequence analysis revealed it is a hybrid between the Neanderthal mother, that is mitochondrial DNA has come from mother, and the Denisovan father, because of the Y chromosome sequences, they could say Denisovan father. So that means at certain stage, there is a mixing up of the Neanderthal individuals and Denisovans. And like this, the exchange of material has taken place, and now this is important. The finding of the first generation of Neanderthal Denisovan offspring among the small number of archaic specimens sequenced to date suggests mixing between late Pleistocene hominin groups. That means during those days, even though small in number, they were mixed together and hybrids were produced. But today we do not have the idea whether these hybrids were fertile, whether the hybrids got to this F2 generation or the F3 generation. The message is they got the F1 generation. They never thought it is an F1, but nucleotide analysis revealed the mitochondrial DNA has come from Neanderthals. The entire nuclear genome has come from both the parents. So this is the first ever hominin hybrid in nature. And with this in mind, you can discuss the concept of biological species concept or the phylogenetic species concept. Of course, this is the three, and these are the bones of the hybrid. At the same time, again, this is in 2020, one more genome of hominin scale was discovered in northeastern Mongolia, and these segments derive from the same Denisovan admixture events between the two different races. That means now Denisovan ancestry is also known, and people know that The Neanderthal genomes and Denisovan genomes are represented in the modern humans, like the Papuans and Aboriginal Australians. So that is on one side. The Neanderthal and modern humans also interbred. These papers have been published. Multiple waves of introgression from Neanderthal and Denisovans have happened. And today we have the humans, and we have the human genome as well as the extinct organic lineages. It was claimed that Neanderthal-like sequences can be identified in every contemporary modern human genome. Today, huge human data is available in data banks, and now people say that comparative genomics tell us that contemporary modern human genome has this representation. Non-African genomes contain nearly 1 to 2% of Neanderthal genetic material. Of course, the locations of the introgressed archaic DNA segments vary between individuals. Altogether, by taking cognizance of the Neanderthal genomes in different Homo sapiens across the globe, they suspect nearly 40% of the Neanderthal genome has been found in. But that doesn't mean that all of us carry 40%. The total amount is there, but this total amount of 40% of the Neanderthal genome is scattered in different individuals in different parts of the globe. One more examples, the list is published. Taking examples of archaic gene variants that influence the physiology of the present-day humans have already been demonstrated. These are some of the implications. I have selected a few for presentation. For example, a clear example of Denisova-derived version of hypoxia pathway EPAS1 gene, which confers genetic advantage to survival in high altitudes, has been found in Tibetans. That means this particular gene, archaic gene, was present in Denisova, and later on, with some modifications, you can see the same sequence is available in Tibetans. As you know, the Denisovans lived in an area, cave, which was filled with snow, and that age was ice age, and they survived in that particular ice age where the oxygen concentration was less. And a similar sequence with some modifications is found in present day Tibetans. Similarly, another example of introgression is a cluster of genes encoding toll-like receptors known to be involved in microbial recognition and reactions. These haplotypes, that is apasia haplotype and toll-like receptors, these haplotypes are present at variable but surprisingly high frequencies in the current human populations. That means two inferences here. Interbreeding has taken place, and now, as you could see, that the apasia gene, what is it doing? It codes a protein. And in mammals, it is a type of hypoxia-inducible factor, a group of transcription factors involved in physiological response to oxygen concentration, and that sequence is present in Tibetans, right? And we have inherited from Denisova with modification. Similarly, the toll-like receptors are a class of proteins that play a key role in innate immune system, and also with certain modifications, all of it. But the frequencies of them depend upon from which populations you are analyzing. So two things here, the hybridization was possible, introgression has taken place, and some sequences are selected, selection pressure has favored them with certain modifications. And descent with modification, and these modified sequences are influencing the physiology of the current day animals. The data from Babo revealed an interesting interbreeding occurred between modern humans, Neanderthals, and Denisovans, resulting in small amounts of archaic genome segments in the present-day humans. And also, as I said, this is a clear example of positive selection, because already they conferred fitness in Siberia, and those same sequences are found in the Tibetans now. And he reported a few sequences which are related to immunity. For example, For example, the chromosome 12, the present day haplotype on chromosome 12, which is associated with, please note this, which is associated with 22% reduction in relative risk of becoming severely ill to COVID infections, appears to be from Neanderthals. A similar sequence was present in Neanderthals. The comparative genomics has shown that particular sequence with certain modifications are located on the chromosome 12 of the present day karyotype. And what is its role? It reduces the risk to the extent of 22% reduction in our risk of becoming severely ill to COVID. And that was published last year. The haplotype is present at substantial frequencies in humans in all regions of the world outside Africa. Again, outside Africa, because in Africa, only after coming out of Africa, the Homo sapiens interbred with the Neanderthals. That's why the populations out of Africa carry this, and they are totally absent in African population, again confirming out of Africa theory. The genomic region where this haplotype occurs encodes proteins that are important during infections with RNA viruses. That means we have inherited a positive gene which improves our fitness, descent with modification, relative selection, pressure, etc. This was published here. A genomic region associated with the protection against severe COVID is inherited from the. And now the most important thing is this. In one of the recent publications this year in Science, they noticed this. The likelihood of a longer metaphase duration and fewer chromosome segregation errors in modern human than in Neanderthal brain development. That means they have studied the modern human mitosis and also what is happening there. And they have compared it. And now the message is that there are fewer errors, segregation errors in the modern human beings when compared to the Neanderthal. They have not studied the mitosis of the Neanderthal, but how did they come to this conclusion? See how they have done, the Pavos group. In the time since ancestors of modern humans separated from those of Neanderthals, around 100 amino acid substitutions seem to have spread essentially to all modern humans. The biological significance of these changes were not known at that time. And now what Pavos group has done is, Pavos team have shown 6 such amino acid substitutions, 3 proteins known to have key roles in kinetochore function and chromosome segregation, and to be highly expressed in the stem cells of developing neocortex, that is brain. So there are many amino acid substitutions. Out of these 100 amino acid substitutions, they have studied in detail the 6 such amino acid substitutions. And of these, 3 proteins are an important role to play in kinetochore function and chromosome segregation, particularly in the stem cells of the neocortex. This was a real breakthrough because, as you could see here, when they have introduced these modern specific substitutions, these substitutions were found in human, and they have introduced these human specific substitutions into mice, transgenic mice. And now these are the genes. And when these substitutions are caused metaphase prolongation and fewer chromosome segregation errors in the apical progenitors of the developing cortex. That means they have a mouse without these modifications. They have a mouse without these modifications in the amino acids, and transgenically they have produced a mouse with these modifications. And they have compared the chromosome segregation. And the mouse which had got these substitutions through transgenesis, they had fewer chromosome segregation errors when compared to the mouse which did not have them. So this is a very clear proof to show that the sequences were present in Neanderthals, but with certain modifications in amino acid substitutions, they were inherited to the human beings, and particularly they play a crucial role in chromosome segregation, particularly in the neocortex development. And they confirmed it by producing a transgenic mice with and without that sequence, which proved that these substitutions, proteins with these substitutions, have a positive role to play in reducing the errors in chromosome segregation. Very crucially, the modern brain is getting developed with fewer errors. For example, these genes, they have studied the roles of these genes are associated with mitotic spindle, kinetochore, and chromosome segregation. These genes where amino acid substitutions have been recognized in human, they are shown to play a role with mitotic spindle, kinetochore, and other segregations. These results imply that fidelity of chromosome segregation during neocortex development likely improved in modern humans after their divergence from Neanderthal. The archaic genes were inherited from Neanderthals, but subsequent modifications have taken place, and these modified proteins have been inherited. And like that, I have selected a few, as I said earlier, that human X chromosome has a gene called dystrophin gene occurred in Neanderthals, but now 9% of all modern human populations outside Africa has this gene. What is that doing? Spontaneous or inherited mutations in the dystrophin gene can cause different forms of muscular dystrophy, a disease characterized by progressive muscular waste. So this gene is inherited from Neanderthals, and it is present to the 9% in most of the human populations. Similarly, there is one more gene which is inherited from Neanderthals that is called BO6. And what is it doing? It is involved in fibrosis, a related gene expression in the hypertrophied left ventricular tissue. That means it is related to cardiovascular disease, and this gene is inherited from Neanderthals. Skin complexion gene from Neanderthals. Deleterious genes also may have been introduced, particularly people suspect diabetes 2 gene must have come from the Neanderthals. And like that, I can go on giving a catalog of genes that are inherited and their impact. But recently, they have shown the genes concerned with hair color, immune system benefits, sleeping patterns, depression, blood clotting, most of these genes concerned with these are inherited from Neanderthals, and now with certain modifications, they are thriving in the human population. And now the last question is, what makes the human human? Access of archaic genomes offers exciting new possibilities to identify critical genetic features that distinguish from archaic humans. And now such a comparison has shown there are about 31,000 SNPs, single nucleotide positions in the genome, where present-day humans from all parts of the world carry only novel derived nucleotides. That the archaic sequences are not there, and only derived nucleotides. That means these genes are inherited, but they are all derived today with certain modifications because of the SNPs. Of these variants that are fixed in present humans, around 3,000 are in regulatory regions, around 30 affect putative splice sites, and around 100 affect protein coding regions, altering amino acid compositions. So these are the things which are making us more human now. They are derived from ancestors, but they are modified, and unique Homo sapien characters are coming up. More and more research is going on now. Elucidating the functional relevance of uniquely human genetic variants is exciting challenge today. And as Pablo says, that is this main research today, which represents one of Pablo's main current lines of research today. I might share, when I was preparing for this talk, I had some email exchanges with this Nobel laureate, and he openly shared some of his, and he also clarified some of my doubts. And he said, as of today, he is more interested in assessing the functional significance of the variants that he has noticed in these fossils when compared to the human beings, and how Homo sapiens are being at an advantage with these modified variations. He was within one day or 2 days, within 1 hour, a couple of hours, he has responded to our questions, and that shows its collaborative approach and cooperative approach. And also he said, I asked him why it is restricted to Neanderthals and Denisovans. Why not to the common ancestor like Homo erectus? Other fossils are available. And his immediate reaction was that the fossils were not there, but those fossils were present in Africa where the temperature was very high. It may be extremely difficult to get DNA, but they are at it. But whereas the Denisovan and the Neanderthal DNA was relatively, they were in a place where they were with minimum damage. It was possible to isolate them, and they continued. Only I am just sharing with you how quickly he responded, and he also said, as of now, this is my research, and I may take it up later. And now, last slide. What is the current scenario of India in this regard? To my knowledge, way back in 1993 itself, Department of Science and Technology organized a brainstorming session on paleobiochemistry at Punjab University. During that meeting, molecular paleontology and molecular archaeology were discussed. And that means we have realized the importance of this study, and the brainstorming session was there. And as I know that once the brainstorming session is organized, they expected some more projects in this line, but I couldn't get more information. What is the outcome of this brainstorming session? Subsequently, Center for Cellular Molecular Biology, under the leadership of Lalji, they started a lab to look at some of these genomes, and now, after his retirement, at his death, and his partner, Dr. Tangraj, is continuing the work, and now he's the director of CDFT, and they are also interested in the ancient DNA research. And one important contribution is from this particular group, Lalji, to start with, is the novel insights into evolution and migration of humans. For example, the fundamental DNA-based research carried by Singh and his colleagues on primitive tribes, including tribal population of the Andaman and Nicobar Islands, provided critical insights into the evolution and migration of humans, suggesting the out of Africa route of modern humans about 60,000 years ago to the Andaman Islands. These findings have furthered the scientific community's understanding of the origin of the Earth. And at present, Dr. Tangraj and his group found the present-day Indian populations are descended from two diverse groups, namely ancestral South Indians and ancestral North Indians. That it was published. And further, they have found that the ASI, South Indian, have admixed during 2,000 to 4,000 years, and it has been published. That means our people, not exactly paleogenomics, and they have collaborated with one of the collaborators of FABO, and they're continuing this. We are not far behind, but it may take some more time to catch up the research in this particular field. One pleasing is our DST has realized, and they had a brainstorming session way back in 1993. When, as I said already, 1984 was the ancient DNA was isolated in a zebra. By 1993, we have realized, but it has not, subsequently, somehow we have not picked it up. And finally, this is the Nobel Foundation picture, and this is the common ancestor, Homo erectus, then Homo hibernensis, then on one line, Denisova, another line. These are all in very protected environment where I say Pleistocene was there. And from Africa, directly, you got the Homo sapiens out of Africa. And later on, these people mixed, Neanderthal and Denisovans mixed, and then the hybridization took place. So this is a tentative figure to show that the physical appearance and so many things are different from Homo sapiens. And many a time people think many of the pictures given in textbooks and papers is they show in one line, all of them in one line, as if it is an orthogenetic evolution. It is not so. there are many side branches. There are many side branches, and many of these side branches are not represented in the straight line. So we have to be careful to see that these are the side branches which became extinct, and one line continued. These people, when they were in Europe, they mixed with them. So here, finally, if Dobzhansky's statement is correct, that nothing in biology makes sense except in the light of evolution, then as we said, it might be appended. Much in evolution makes more sense in the light of phylogeny. What all changes people got, what all information, what are the lessons they learned from paleogenomics of Sabo has not only helped us to understand, and also it has enriched our phylogeny. And that's why he amended it. And now that is the message is the paleogenomics have made us to access the fossils region, which was inaccessible to all of us in the early days. So not only morphology, not only distribution, not only the characters, even this DNA could help us to learn more. And we all of us share, Homo sapiens share the representations of the Denisovan and the Neanderthals, which are no longer present here, but they live with us in some form or another in our genome. So finally, through his groundbreaking discoveries, Svante Pabo opened a new window which was not available to us to understand our past, revealing an unexpected complexity in the evolution and admixture of ancient hominins, as well as providing the basis for an improved understanding of genetic features that make us uniquely human. So two things were asked earlier days: how are uniquely different from Denisovans? And at the same time, the converse was how then Neanderthals were uniquely Neanderthals, not human now. So finally, once I said to them that modern Homo sapiens are glorified Neanderthals because we have a mixture of both, and with certain modifications, we are surviving today. So thank you very much. This is in brief I wanted to share with you people. Thank you very much.
Thank you very much, Professor Ranganath, for a very illuminating talk on this year's Nobel Prize for Physiology or Medicine given to Professor Svante Pabo. ನೀವು ತುಂಬ ಚೆನ್ನಾಗಿ ಎಲ್ಲ ವಿಷಯಗಳನ್ನು ವಿವರಿಸಿದ್ರಿ. While talking about the contributions of Professor Pabo, you also have brought before us the entire evolution, human evolution before us in the last 1.5 hours with illustrated slides. And the talk assumes greater significance since you had a first-hand contact with the Nobel laureate this year. The mails that you have exchanged with him bring so much of the first-hand information to the talk today. We cannot be grateful enough for the kind of information, knowledge, and the analysis that you have made with regard to how the whole evolutionary sequence has taken place. That also shows your in-depth knowledge in the subject and your passion for the subject with your lifetime experience. So from the Karnataka Science and Technology Academy, we are grateful to you at this stage, since we have very illustrious audience also with us. If there are any suggestions, comments, questions, you are welcome now. Open to the audience now. Are there any remarks from the audience?
There are two questions from the audience.
Yeah.
Before unmuting them, I would like to take suggestions and comments from other panelists. That will be
Yeah. Are there any? Yeah, we can go to the questions, please. Hello?
Sir, yes, sir. The human evolution from Neanderthal hominins, human, is it right, sir? There is a question from Dr. Tippeswamy. I would like to unmute them. They can directly speak to sir.
What is it?
Dr. Tippeswamy, please.
Yeah. What was the question?
Dr. Tippeswamy, you can unmute and speak.
Dr. Tippeswamy, sir?
Sir, I'm audible, sir?
Yes, sir.
Yeah.
Yes, sir. Please.
Yes, sir. Sir, Ranganath, sir, ತುಂಬ ಚೆನ್ನಾಗಿ talk. Sir, regarding evolution, ನಿಯಾಂಡರ್ತಾಲ್ ಹ್ಯೂಮನ್ ಎವಲ್ಯೂಷನ್ ಆಗಿದೆ ಅಂತ ಹೇಳಿ, ಮತ್ತೆ ಈಗ ಹೊಸದಾಗಿ ಐಡೆಂಟಿಫೈ ಮಾಡಿರ್ತಕ್ಕಂಥ ಹ್ಯೂಮನ್ಸ್, ಸೊ ದಟ್ ಈಸ್ ದ ಮಿಡ್ಲ್ ಆಫ್ ದ ನಿಯಾಂಡರ್ತಾಲ್ ಅಂಡ್ ಆಲ್ಸೋ ಹ್ಯೂಮನ್ಸ್.
Tippaswamy,
ಸರ್,
Tippaswamy, voice is not clear.
ಓಕೆ ಸರ್.
Okay, Mr. Umesh,
Yes,
you
sir.
can read out the question.
Sir, the isolated mitochondrial DNA and genomic DNA from the fossil finger bone, how they have isolated?
That's why I told you, no, I have not gone into the molecular details of how the extraction of them. That takes a lot of time to give. One of those biologists who are sequencing, they could enlighten you more. But what I am interested was the outcome of these findings.
Thank you,
Thank
sir.
you, sir.
There
What
is
is the
one
next
more
question?
What is the
Dr.
utility
Muralidhar
of-
has asked a question. What is the utility of phylogenetic species concept? Dr. Muralidhar, sir, biological species concept, as I said, emphasizes on reproductive isolation, and virtually it may be difficult to establish the species based on reproductive isolation because of technical difficulties. Therefore, contrary to that, as long as you believe in descent with modification, we have the ancestors with certain modifications have given rise to the derived ones with certain modification. That means the phylogenetic species concept emphasizes the phylogenetic lineage rather than reproductive isolation. So earlier, the Denisovan and the Neanderthals, they were treated, in some reviews, they were treated as races. Some people have treated them as subspecies. But now, if you extend the phylogenetic species concept, phylogenetically they are different. Uniqueness is there. Denisovan uniqueness, the Neanderthal uniqueness, as well as the Homo sapien uniqueness. Based on the phylogeny, based on the descent, based on the uniqueness, they deserve to be treated as different species. That is the beauty of phylogenetic species. Biological species concept very rarely emphasizes on the phylogeny. It only parameter benchmark is isolation. But here, the uniqueness, the diversity, what you see in nature of the species is because of the phylogenetic inheritance, and at every stage, one cannot explore how to establish whether they are reproductively isolated or not. Is it okay, Muralidhar?
Yes. Yes, sir. Thank you, sir. Thank you so much. As I said, yeah, Dr. Tippaswamy, you want to say something?
No, can I say something?
Yeah,
Dr.
please.
Shivanandappa, please.
Yeah. I should compliment my friend Professor Ranganath, who is the right person to talk on this subject, having taught genetics and done research in the area. And he has very extensively covered in depth the group led by above. But some of the things which I am also aware of and in touch with the recent developments, very illuminating. And we never thought, as he said, that evolutionary biology will never get the Nobel Prize. And this is such a great welcome thing in the world of science, where evolutionary biology has referred to humans has illuminated our past, because evolutionary biology, as refers to other species, would not have captured the attention of Nobel Committee, because it is the humans and the human society, because the implications of this research has come even to populations such as in India, where we have tremendous variety of caste population. Each caste in India is a genetic unit, and it offers tremendous opportunity to know us about the diversity of the population. So, as he has already referred, Professor Thangarajan in Hyderabad, who has collaborated with both David Drake in Harvard and Pabo, they have extensively studied to tell us about how different we are, but we are really not truly different. There is so much of hybridization before the caste system evolved, where isolation mechanism, reproductive isolation mechanism led to reduction in the variability as would have happened much earlier. So therefore, the implications to the human society are immense. And I think this is an area which makes us really realize the importance of studying the genetics and biology, which should be taught to students at the elementary school, so that after all, we are not so different. We all are one. So I think that's the message that one can give as a sociologist. Thanks, Professor Ayyappan, for organizing these things, and Professor Ranganath, who has put in tremendous effort to make this lecture a very uh, listenable, interesting topic for anybody, not necessarily experts. Thank you.
Thank you, Dr. Shivananda for these comments and a valuable addition. We have the last question from Dr. Hanumantappa, I believe. He asks the question like this: may I know some of the research questions in this field where I can integrate with machine learning algorithms? Professor Ranganath, sir.
Yes, yes. Today, physically, it is extremely difficult to compare the information that is coming from various sources. So artificial intelligence is one which can mine the content of the genomic sequences. And if you have a new algorithm with artificial intelligence, you can extract more information from these things. It is up to you, up to the people to develop new questions, accordingly new softwares to look into it.
Thank you, sir. Thank you very much.
There is a question
As I
for
said
Dr.
earlier, we have no words to thank you enough. Such a detailed analysis of the entire subject and such a lucid presentation that we have had. And as I said, this will be also on the YouTube for benefit of the much larger audience from the Karnataka Science and Technology Academy. Grateful for the first lecture on the Nobel laureates in 2022. At this time, I also have my colleagues, Dr. A.M. Ramesh, CEO of the Academy, Dr. Anand, Senior Scientific Officer, and Shri Umesh Gatge, the Scientific Officer with us. I'll now hand over to Dr. A.M. Ramesh, CEO of the KSTA, for his words of thanks and his words. Dr. Ramesh, please.
Thank you, sir. Good afternoon, everybody. Sir, the Nobel Prize Lecture Series 2022 was started from today. This series, first talk on Nobel Prize in Physiology of Medicine, was delivered by distinguished scientist, renowned academician, and esteemed member of KSTA, Professor H. Ranganath, sir. As our Honorable Chairman summed up, the talk covered evolution from the concepts to the Nobel Prize-winning research of Swedish scientist Svante Pääbo on his discoveries concerning the genomes of extinct hominins and human evolution. Sir, it was a very scholarly and enlightening talk, which even a graduate student could understand. And the one take-home message from the Nobel Prize-winning research was collaborative research that Pääbo initiated. It was a great thing. And sir, on behalf of KSTA, I thank you immensely for this extremely informative and scintillating talk. And also my special compliments for your PPT, which were very clear and we could easily understand what you were talking. I am also grateful to our Honorable Chairman, Professor Ayyappan sir, for his guidance and helping us in organizing this webinar. I'm also thankful to our esteemed members of KSTA and also Dr. Ramakrishna sir and Dr. T. Shivananda sir for joining the talk. And I also thank all the academicians, scientists, and students and my colleagues who have joined for this webinar. I would like to thank the viewers who have joined on Cisco WebEx and also YouTube. And as our Honorable Chairman told, those who have not joined could also view from YouTube that we'll be uploading. With this, I am very thankful to everyone who have joined. And tomorrow, Dr. Ravi Shankar C. Kodihalli, the Senior Principal Scientist and Associate Director, Pfizer USA will be delivering a talk on the chemistry Nobel Prize research by Caroline Bertozzi, Morten Medal, and Barry Sharpless for their on their development of click chemistry and bioorthogonal chemistry. I request all of you also to join tomorrow and day after tomorrow with this. And once again, thanks all the viewers on Cisco WebEx and YouTube. Have a great day. Thank you very much.
Thank you, Ramesh.
Thank you. Thank you, Dr. Ramesh.
Thank you, sir.
Thank you, Professor Ranganath sir. Thank you very much for your guidance and the present talk. ತುಂಬಾ ಧನ್ಯವಾದಗಳು. ನಮ್ಮೆಲ್ಲರ ಕಡೆಯಿಂದ ಧನ್ಯವಾದಗಳು. ನಮಸ್ಕಾರ.
ನಮಸ್ಕಾರ.